The lesson that we're going to look at today is lesson 4 of our biology, homeostasis, and type 2 diabetes curriculum. And I was saying it had been out a while, but as we're getting sort of to the end of this grant and wrapping things up, I think that it has some really important concepts that haven't been maybe highlighted, or we haven't done PD on this lesson specifically, and I just wanted to make that happen. All right, so here we are teaching about gene-environment interactions. I will take a minute to shout out SEPA, the Science Education Partnership Award, which funds all of our work here that we've done over the last 5+ years on this grant. So when I think about how we biology teachers, and not just biology teachers, but also in sort of the general public, we talk about genetics in a way that quite often uses single gene traits, or what are called Mendelian traits, that sort very nicely using single genes to show how genetics works. And we reinforce that using Punnett squares, and we use pedigree charts and things that are very kind of simple and easy. And I will say, as a biology teacher, I love teaching genetics for this reason. It seemed like it was able to take a lot of complexity, and you boil it down into a nice little Punnett square, boom, 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 you've got your ratios, it all works out, it's really nice and mathy, and it works great. But the more I've been doing this, and especially now that I'm in a department of genome sciences, I realize that at the level of genomic research, that's not really how genetics is talked about or presented. And so I become a little bit more worried about the way that we use these single gene traits to teach genetics. And then we quite often reinforce it with things that aren't single gene traits, like eye color, for example. They're not just 2 genes that contribute to eye color, and we use those for Punnett squares. I mean, maybe you don't. I know I did in the classroom. I would use eye color as an easy way to talk about Mendelian traits. And I'm poking fun at ourselves because this genetic trait sheet that I just put up here is produced by Genome Sciences Education Outreach. This is from our very own curricula from a number of years ago. But these things, attached earlobes, tongue rolling, mid-digital finger hair, these are not single gene traits, and we shove them in this package so that we can use it to teach about Mendelian ratios and the things that we think that we need to teach. But in the end, I think this is really doing a disservice, because quite often we're driving the message of the 1 gene, 1 trait message, which is not true. And then we go on to not only work with these misconceptions, but then tie them to race in a way that's just not true. So we talk about sickle cell anemia being a Black disease, or cystic fibrosis being more common in the white population. So what we're doing is driving some misinformation, and there's some narratives around race that are just false, that really leave people with the idea that, it kind of drives genetic predeterminism. And you hear this in the culture a lot, where you hear the word the gene for, the gene for this or the gene for that. And that it's so much more complicated for that. And even if you're a FACS or a culinary teacher, you end up teaching a lot of health concepts in your classes as well. And so I would just caution you, every time people go, oh, well, it's genetic, not to just stop there, because that's very rarely true, with the exception of some very specific genetic traits. And it's not true at all with the sorts of diseases and health conditions that really impact students and their families. So if we're talking type 2 diabetes or cancer or heart disease or high blood pressure, all of those sorts of conditions are not driven primarily by genes, but also by environment. So instead of Punnett squares and pedigree charts, I'm going to throw this out there, and we'll come back to this chart. But when you're talking about genes, conditions that have very many genes that contribute to that condition, you are talking about allele combinations. And what we've done in the past is really to focus on two. I will back up here. We have 2 suggestions for you to help teach this more coherently. And the first suggestion is to start with the big picture. So really perhaps start with population genetics rather than individual genetics. So most of our traits are polygenic, where you have many gene variants that contribute to the condition. It's not so much a matter of recessive and dominant genes, which is the storyline that gets told a lot, but more of how many risk gene variants you have and how many non-risk genes you have. And this is often shown in the shape of a distribution curve rather than you have it or you don't have it. So as I say, we're going to come back and dissect this chart a little bit more, but this is kind of the idea where we're talking about distribution curves, a lot of people being sort of in the middle, a few people being outliers on the sides of the curve, depending on how many on their genetic factors. So genes do make a difference. I'm not saying that they don't, but there are a lot of other things that contribute. So our second suggestion to kind of move away from the single gene trait story is to expand the definition of what the environment means. So quite often in biology classrooms and in textbooks, if you're talking about the environment, and if you go to the index and look up the environment, it talks about temperature and pH and humidity and light cycles. And or if you're lucky, like the presence of mutagens might change your DNA, which then will change the resulting traits. So this is all true, but the environment is so much broader than that. And so it would be helpful to really look at a much broader interpretation of the environment and figure out how things like access to parks and sidewalks and grocery stores and fast food chains and transportation also impact an organism's health beyond temperature. So there are a lot of drivers of health that are not genetic that are hugely important. You know, if you're talking about transportation, can a person get to a doctor's appointment? I mean, that in some ways has much more impact on health outcomes than the number of risk genes or non-risk genes that they may have. So for our curriculum, we use type 2 diabetes as our driving phenomenon, because as I mentioned before, this could work with cancer, heart disease, high blood pressure, you name it. There's some bit where this can work together, where you're talking about polygenic traits, so traits that are determined by more than one gene, but a whole host of genes. And then they're also impacted by the environment. So when you're looking at data like this, and both of our curricula start with these wide-ranging pictures, you can't explain it through pedigrees and Punnett squares. And in fact, people have been asked, there have been a couple of diabetes studies where they will ask people, why do you think you've been diagnosed with type 2 diabetes? And folks will say, well, my grandma had it and now I've got it, so maybe it skips a generation. And you can see them trying so hard to use what they've learned in high school biology class to make it all make sense, but they're building on the wrong framework. So when students can see trends in diagnosis rates and these changes over time and geographical differences by introducing the population level data first, they realize that they can't solve it in the way that they have been taught perhaps so far. And we're working with a multifactorial trait, not just a simple Mendelian trait. So when you look at a map like this and you see that there are different rates of type 2 diabetes, or this is actually diabetes as a whole, but a very large percentage of all diabetes diagnoses are with type 2 diabetes, that you see that there's changes, geographical differences, things that can't be explained. So you can see that it's not necessarily driven by changes to our genes. So if genetics were the only driver of diabetes, why do these geographical disparities exist? And how can it move so quickly? So if we're talking about the last 20 years and a real huge amount of people being diagnosed with type 2 diabetes, that doesn't sound like a genetic disease per se. Genes take generations to pass on, and it's something about a change of our environment that is impacting or unmasking genetic susceptibility for large numbers of people over time. So what we're trying to do is shift the genetic storyline from examples of single gene traits to examples of multifactorial traits. And when we do that, we also are shifting the target for solutions, because once you say, well, something's a genetic disease, there's not much you can do about that. But when you're talking about environmental solutions, then suddenly there's a whole world of things that students can work on together or become aware of to fix some of these problems. So lesson 4, which is what we are going to do today. With that introduction, I'll get off my soapbox now. I've been getting higher and higher on this soapbox, the more closer to retirement I get. The one thing I want to tell people. So lesson 4, the essential question is, how do environmental and genetic risk factors influence a person's risk of developing type 2 diabetes? And you could switch out type 2 diabetes for many other disease models if you would like to study something else. And then these are the big ideas. I will let you look through those. And again, a focus on prevention, because when you're talking about environmentally mediated conditions, then prevention, suddenly there's something that you can do about it. And it's a type of prevention that goes beyond individual, like the story that we hear all the time, which is not untrue, but it's around personal choices, like exercise more and eat better. And those things are true, but there are also some environmental conditions that really weigh the scale, tip the scale one way or another. And the place policy in type 2 diabetes is really going to dive into that, this idea of how access, your choices are really nested within what you have access to. So that's in 3 weeks. So come back for that if you're interested in that. So what we're going to do now with the rest of our time is go through the lesson. And the first thing you're going to do is simulate your own genetic risk score for type 2 diabetes. And then after that, you'll have some time to determine your own environmental risk factor using our handy dandy survey. And then we're going to come back and put it all together and look at that distribution curve again and make some sense of that. So if you're doing this in the classroom, this is a bag full of beans, and you put the same number of, in this case, white beans and red beans together, and that the bags in the bean are the gene pool. Students are going to pull out 2 beans at a time. Those 2 beans represent the alleles that they inherit from their parents, and they drop 2 beans into each of the 12 spaces in an egg carton. And based on what they randomly draw, they're going to be simulating their risk. So if you draw 2 white beans, it's a positive risk. 1 of each is neutral, and then 2 red beans is a protective. It decreases your risk, so you give yourself a -1 for that. So this is a good time to talk about some of the math that goes along with this. If you know that your gene pool is a 50-50 mix of these different colors, so what would be the probability of drawing 2 white beans? I know you want to say 50%, but it's a 50% for the first bean and 50% for the second bean, which makes a 0.25 or 25% chance of drawing 2 white ones together. A combination, you could draw a red and a white, or a white and a red, which is 50% chance to draw a neutral, and then 25% chance to also draw the protective red risk factors. And if you want to introduce the terms or refer to them, it's a great way to do it. And yes, this looks like it could be a Punnett square, because it could. Punnett squares work for a reason, but we're not talking about it in terms of dominant and recessive. We're talking about in terms of allele, types of alleles. One confers a higher risk, one a lower risk. All right, since we don't have bags of beans in front of us, I've asked you to have 2 coins with you, or you could use 1 coin and flip it twice, but however you want to do that. So what I would like for you to do is go to student sheet 4.1, which is on here. It's also on the workbook, and I'm going to give you 8-ish minutes to work through that. So you're going to be flipping a coin, figuring out your risk, whether you have a +1, a 0, or a -1 for each of 12 times, and then you'll record that on a communal distribution chart. Any questions about that so far?
You said half the beans are white and half are red. Is that a somewhat accurate reflection of the allele frequency in the US population?
Absolutely not. So,
I didn't think so. I just asked him.
Well, it depends. So after we'll get to that after we do, this is sort of the base exercise, and then we'll talk about allele frequencies in the population. And is one of the ways that the model is not totally accurate. So thank you. Great question, Jamie. All right. So let me see if I get the workbook up and student sheet 4.1. So it is, you know, you've got your columns of pluses and your columns of minuses. It's kind of like card counting where they cancel each other out and then you figure out what you have in the end. So if we look now at our, actually, just while we're all here together, are there any questions about just that portion of the activity, sort of from the, have any of you done this in class already? Okay, great. All right, well, I'm going to go to our, we have a few sticky notes on here. I'm going to go back to, I faked this one, whoops. Sorry, every time, got to go back for this. So this is one that I faked it before, but generally with a number of people in the class, and especially if you do this with more than one class and people just keep adding to it, you end up getting a pretty random distribution curve. Some people will have randomly high numbers, some will have low, a lot in the middle, and the more data you can add to this, the more likely you are to get that nice distribution curve, especially with the 50-50 mix. Now, it doesn't just stop there, though, because if you're in the classroom, it's really nice to have a bag or two that's made of a high-risk population and a low-risk population where the allele frequencies are not 50-50, but you might throw in 2/3 of a cup of one and then 1/3 of the other. So if we want to simulate a high-risk population, you could do this. It's nice not to tell the students that some of the bags are different. So they're all doing it. They come to this end where they start doing their distribution score, and there might be few that are way out there, and they're like, well, wow, why do we have such low scores or such high scores? And then you can kind of do the big reveal. Well, look to see what the distribution list is on the bottom of your bag, and you hold it up and like, oh, well, I had a high risk population, which just means that the allele frequencies are not the same. So then you can go through the math with this, and it's a much easier to get, say, 2 white beans if you've got 3/4 or 2/3 of the beans in the bag, if you spike the bag a little bit. So the idea here is that what this shows is that we're looking at the scores that you get with only 12 genes. So that's what's in your egg cart in our 12 little spots, and that's what we're going to do. And if you do have a 50-50 mix, which is not the case, there are many gene combinations, and they have all sorts of different frequencies within the population. But let's just say that everything's 50-50 for the sake of argument. What would the chances being of only drawing white beans for this entire all 12 of these? And you realize that the chances of doing that, the probability of doing that is exceedingly rare. It's not going to happen. Well, yeah, you have a 1 in 16.777, 216 million chance of that happening, to just happen to draw all white beans. But then when you start thinking about the fact that there are over 400 genes that are associated with type 2 diabetes, and actually thousands that people are looking at to see about the correlation, 400 are pretty tightly correlated. We're talking about such a large number of genes and the large number of probabilities that can happen, that it's this huge number of combinations that leads to the tremendous variation in traits in a population. And that's one of those DCIs, I guess, it's kind of hard to get to in my mind, but this activity kind of brings it all home. Like, well, you've got hundreds and hundreds of genes that we're talking about. And then at the bottom here is the limitations to the model, which there are a few, and that each allele associated with type 2 diabetes is present in the population in the same frequency if you don't use the high and low risk population bags. And there's more limitations than that also, but there they are. And you would have to return the beans to the bags every draw to be totally accurate. But I did run this through a population geneticist, and she said with that number of beans, it should be statistically a wash. Don't worry about returning the beans to the bag. Okay, so that is our genetic simulation. The idea is to show that there are a lot of ways to get variety, to show how that distribution curve gets built using a number of variations of alleles over a number of different genes that might contribute to a population or to a condition. Type 2 diabetes, not unlike a lot of the big multifactorial conditions, there's no one smoking gun gene. There's no one gene that pops its head up above everything else. And you can say, if we could just fix this one genetic cause, we would impact a lot of type 2 diabetes. It doesn't work that way. And it's the same is true with cancer in most conditions, in most cases. There are some, of course, like the BRCA1 mutations that do lead to a higher chance. So that's the genetic explanation. And now the next thing we're going to do is do our environmental simulation. And there are a couple of ways to do that. This is the old-fashioned pen and paper one that's in the curriculum that students need a little help. Sort of, you read the first line, number of fast food establishments in your community meaning within, I think, a half a mile of your house, we decided. Me, I'm in the middle of Bellevue, that I've got lots. So I've got more than 4 in front of near my house. So I would have a +1 score after circling a 4. We also have a much easier and more fun to do is a type form where you can do it online. So I'm going to give you 6-ish minutes to do this as well. And let me drop this into the chat. It's also on our workbook.
That was plenty, but the gifts were great. I did it.
Adam picked all of those out. Yeah, those are pretty awesome. Okay. Great. Well, we will move on. It's a little tricky because you're doing a total simulation for the genetic portion, and then for the environmental portion, it really is your own life. So just be thoughtful about students wanting to share or not share their genetic or their environmental risk scores, because that's sort of their information to keep or share as they want, or do it anonymously if you would like. So what I'd like to do now is, does everybody have access to the workbook? Okay, or somebody, we're gonna go into groups and talk a little bit more about the chart. So if you can see this, and then we're gonna go into breakout groups, and then there are, I want you to just to really sit with this for a minute, make sure that you really kind of understand the X and the Y axis, think about questions that your students might have about what's going on in this. And then there are 3 questions here on the side to discuss as a group. So can a person without many risk alleles get type 2 diabetes, or a person with a lot of risk alleles not get it? Anyway, you see the other 2 questions here. There are a lot of overlapping area. And then, is this graph only work for type 2 diabetes? All right, so just to finish up by saying that hopefully there's a lot more clarity around this sort of showing genetic material in this sort of graphic, to get away from the Punnett square sort of model, and think about allele combinations and risk alleles and protective alleles. And then thinking about the genetic and the environmental component of getting a disease like type 2 diabetes, where you might be person A has a very low genetic risk score, but a huge environmental risk score, which puts your risk over here in the red. Or you already have a high genetic risk score, and then your environment can sort of pull you back if you have a very poor genetic risk score. And so the end of this lesson, I'm going to skip about that, looks at there are two pieces of two graphs which you could spend time with if you wanted to, but it really looks at people in different risk categories for type 2 diabetes based on the number of risk alleles that they have. And this study put people into 4 different groups of lowest genetic risk score to highest genetic risk score. These are people who had pre-diabetes, so this is the diabetes prevention program. They were randomized to either a placebo group, metformin, which is a common type 2 diabetes drug, or lifestyle changes. And then they just followed them for a number of years to see what their outcomes were. And you can see that the thing that has the most impact for everybody is the white bar, the lifestyle changes, independent of your genetic risk. So this was something. And then just quickly, I'll show you some more data which looks at 3 different allele combinations and how that impacts their incidence of type 2 diabetes, whether they're on placebo, metformin, or lifestyle changes. So that's it for that.
So what?
Yes.
Lifestyle changes had a much higher effect than medication. Is that what I saw there? Or?
That's what you saw there. That is correct.
Huh.
That is correct. So the questions to ask students as you're wrapping this up is, back to the genetic risk score. Does knowing your genetic risk score impact your behavior? So if you know that you're really high risk or really low risk, and that's a really rich question just right there, because none of us really know our genetic risk score. We know our family histories, which is helpful. But how should we be acting? What should our behaviors be as if we're a low or high genetic risk? We can tell about our environmental risk scores somewhat, and that's something that we can also do something more about. So that makes a difference. There's one question I just want to, I know we're over time, on a student handout that tends to be confusing. So if you use this, I just want to give you straight from the horse's mouth here. The question is, does a person's environment change the genes for diabetes in the egg carton? So you've got your egg carton, you've distributed all of your genes, you've gotten your risk score. And the question is your environment actually changing the nature of those genes? And with some conditions like cancer, for example, you might have external mutagens that are changing the genes in the egg carton themselves. But with type 2 diabetes, the environment is sort of pushing your environment. It's changing the way that those same genes are expressed. So changes in our environment are probably more than likely changing the expression of certain genes. And so we're not going in there and changing the DNA of the genes, we're changing the expression of the genes. And that's a whole new, I would love to see more things in biology classrooms sort of get into that, because that seems to be where a lot of the action is. And it takes us away from, you have the gene or you don't have the gene. It's like, well, what's the expression of the gene look like? And what are the things that are pushing the expression of that gene, and how changes in our environment in the last 20 years really driven this change in diabetes and cancer and a number of different things. All right, at the bottom of the worksheet there are ties to the NGSS. If you want to know more about that, there's links to the lesson, the old PDF of the lesson. This is all changing in the next month or so. Up on our website, there will be sort of a new format of this, just FYI. And then there's a session evaluation. It's super short. We would love for everybody to fill it out, but especially if you need clock hours, if you're looking for a STEM clock hour, then you do need to sign in and do the session evaluation. Okay, any other questions? I don't want to keep you any longer on a Monday afternoon. So thank you all for joining us. Really appreciate it. And maybe we'll see you next week too. And Adam, we'll talk about our blood sugar balance game, which is a load of fun, and it'll be a good time.